This is part 1 of 3 on chapter 11, and we're going to be looking at the physical and chemical agents for microbial control. So we're going to be looking at different ways that we can either slow down the growth of microorganisms or to actually kill these microorganisms. So we're going to be looking at decontamination, and this is when we use a physical, chemical, or mechanical method to destroy microorganisms or to reduce the undesirable microorganisms in a given area. So we're going to look at these different types of decontamination. And usually when we talk about decontamination, we want to target microorganisms that are capable of causing infection or spoilage. So any microorganism that can cause an infection, like a disease in a person or animal, or spoilage of food of any type. So that's what we're trying to decontaminate, trying to get rid of those microorganisms. And these things, we're not just looking at bacteria, we're looking at bacteria, the fungi, the molds, the different protists. Some of them, decontamination methods will take care of viruses, and some will take care of the parasitic worms. So microorganisms have different levels of resistance to decontamination methods. So microorganisms that have the highest resistance, these include the prions, which remember, prions are misshapen proteins, and then also the bacterial endospores or spores. Remember, spores are produced by bacteria when the environment gets really bad, and then these spores help those bacteria survive those harsh conditions. So these two things have very, very high resistance. It's very hard to get rid of them and to kill them. Other organisms have a moderate resistance. So these include certain types of bacteria. So the Pseudomonas bacteria, Mycobacterium tuberculosis, which causes tuberculosis, Staphylococcus aureus, which is found in a lot of skin infections, and then also the protozoan cyst. So cyst, they're kind of like spores. They're the form of a protozoan, an animal-like organism that is used to survive harsh conditions. And then a lot of microorganisms have the least resistance. So these are the ones that are easy to control, easy to kill. These are most of the bacterial vegetative cells, so the living, metabolically active bacterial cells, fungal spores and hyphae, so any fungal type of infection, that includes yeast. Viruses that have an envelope to them, they have a lower resistance. And then the protozoan trophozoites. Those are the protozoan part of the life cycle. That's the active, metabolically active site, the metabolically active form, or the form that reproduces. So we have microorganisms that can have different resistance to these different decontamination methods. So this is kind of a summary of this chapter, this image here. So we have these microbial control methods. And we're going to be looking at the physical agents, which are in the pink on the left-hand side. So we're going to be looking at different types of heat and then also radiation. We're going to be looking at chemical agents. These are either gases or liquids, so some type of chemical that we can use to slow down or to kill microorganisms. And then there's also mechanical removal methods. We're going to be looking at filtration as one of these methods. So we're going to kind of follow this flow chart here. So it'd be a good idea to have this in mind as we go through them. So different types of terminology that are going to show up as we go through the different types of decontamination methods. So some methods are going to sterilize, so we're going to have sterilization. Sterilization is when you destroy all viable microbes, including the viruses and endospores, those forms that have very, very high resistance. So basically sterilization is you're going to destroy all the living things, including viruses, on your object that you're trying to sterilize. Other methods are going to be called disinfection methods. So this is a process where we destroy vegetative pathogens. It does not destroy the endospores, and it's usually done on inanimate objects, so non-living things. These include Bactericides, so bacteria sides are chemicals or methods that are used to kill bacteria. Fungicides kill fungi. And then antiseptics, these are disinfectants that can be applied to exposed body surfaces like skin, for example. These methods include like hand washing or iodine, which they use to clean the area before they do injections. Another one is hydrogen peroxide, is used to disinfect body surfaces. Other terms that we're going to see include sanitization, which is where you're going to use any technique that uses a mechanical method to remove microorganisms, debris, or toxins from an object. This includes like a dishwasher, for example. And then degermination, this is just a method that reduces the number of microbes. It doesn't get rid of all of them. And it does this through mechanical means on living tissue. So like alcohol wipes or soap and water is an example of this type of method. So microbial death, it's really hard to detect when a microorganism is dead because they don't have things like a heartbeat, they don't breathe. So there's no vital signs that tell us that a cell is dead. So we define a microorganism as being dead when it can no longer reproduce. So the permanent loss of reproductive capability, even when that cell is placed in optimum growth conditions. If that cell does not reproduce, it's considered to be dead. So other factors that can affect death rate include the number of cells on the object. So the more cells that are on the object, the longer it takes for the object or the liquid to become sterilized. And this is like if you have a high load of microorganisms, it takes longer to sterilize it than if you have fewer microorganisms, it won't take as long. So that's one factor that can affect the death rate. Other factors include the type of organism that is living on the object. If you have spores, it's going to take longer to get rid of the spores than it is to get rid of the vegetative or the metabolically active cells. You can also look at the type of agent that's used. So a microbistatic agent, it just slows down the growth. Or if you use a microcidal agent, that's actually going to kill your living organism that's on your object that you're trying to sterilize or disinfect. So there's lots of different ways to control microorganism growth. And the type of method that is chosen depends on different types of circumstances. So in some cases you might use filtration, other cases you might use UV light. In other instances you might use some kind of chemical to disinfect the skin. So the selection of your method depends on what you're trying to sterilize and a bunch of other things. So here's different questions you should be looking at before you pick your method. So does the application require sterilization? So do you have to sterilize the object that you're going to be working with, or is it okay just to disinfect it? Is the item going to be reused? Can the item withstand heat, pressure, radiation, or different types of chemicals, or is it going to be destroyed by the heat or the radiation or the chemicals you use? Is the method suitable for the circumstances? Will the agent penetrate to the necessary extent? Is it going to sterilize the object when you need it sterilized, or is it just going to disinfect the object? And then also, because in the health field or the veterinary field, you need to look at the cost of the method, and then also, is it labor efficient, is it worth the time, and also, is it safe for the object or for the patient that you're using this method on. So that's why there's so many different ways, so many different control methods we're going to be looking at, and the one you choose depends on what your circumstances. So these different agents that we're going to be looking at, these antimicrobial agents, they destroy or slow down microorganisms in different ways. So these physical or chemical agents we're going to be looking at in the next part of this chapter. Some of these agents will destroy the cell wall. So some chemicals, they cause a cell wall to become fragile, and if your cell doesn't have a cell wall, it could cause a cell to lyse or be destroyed, just break apart. And if you no longer have a cell wall, the cell lyses, it breaks apart, your cell is no longer considered to be living because it can't reproduce anymore. So some examples include antimicrobial drugs, so like penicillin targets cell walls, detergents destroy cell walls, alcohol destroys cell walls. Another method is that you can destroy the cell membrane. And if your cell membrane falls apart, it loses integrity, your cell is going to die, it's going to fall apart. And examples of methods that work this way include detergent surfactants. So these detergent molecules, they look very similar to phospholipids. So if you remember, phospholipids make up cell membranes, and the phospholipids are little brown circles with the two fatty acid tails in here. So detergents, they look very similar, but instead of two fatty acid tails, they just have one. But they still behave the same way. These detergents, they're going to insert themselves into the plasma membrane, and then that causes the plasma membrane to break apart and lose integrity. Other modes of action include number 3, proteins and nucleic acid synthesis. So there's different physical or chemical agents that will prevent DNA replication. It'll prevent the transcription translation, so using that DNA, transcribing it into an mRNA, and then translating that into a protein. Other agents prevent the polypeptide from being formed, so the peptide bond between the amino acids from forming, so it can just stop the protein synthesis. And examples of these, chloramphenicol, which is a antibiotic, UV light works against nucleic acids, and then formaldehyde is another example. If the proteins are created, we can specifically target the proteins, so we can disrupt or denature the proteins, so just cause them to break apart so they can no longer work. So things that do this include alcohols, phenolic compounds, acids, heat. Heat's a really good one for denaturing the proteins. And here, it's an example of a protein. So up at the top we have a protein in the native state, so it's all folded up into its nice form. If you change the heat or change the pH, you could cause the protein to unravel, so denature, like on the left. In the middle, we cause the protein to change shape. Heavy metals, for example, will fill in the active sites of your protein, so your protein can no longer work. So you can specifically target proteins, shown in this slide.